Hi there, welcome to this video. So this is 5.1.1F, which is the specific immune response. Now there's three parts to this spec point. I've done an overview, which you probably want to watch before this one. And then this one is about the T cell response. And then there's a separate video for the B cell response. You can hear my little kitten meowing in the background, sorry. She's about to jump on my neck, so I'll probably jump. So you might have watched my previous video, and so I've got this little way of remembering it where I say tea and cake, and it just reminds me that the T cells are involved in the cell mediated response. If you don't know that already, please go back and watch the other video. So this video is all about the T cell response, which is our cell mediated response. And what that means is that we've been infected by a pathogen, and the pathogen is inside our cells. That's why it's called cell mediated, because the pathogen here is inside one of the host cells. So that little red thing is the pathogen, and the black line is the host cell. Okay, so what first happens here is that this host cell that's been infected marks itself as an infected cell. Otherwise, your immune system wouldn't know that the cell is infected, has been infected. So what it does is it gets the little antigens from the surface of this pathogen and it presents them on its own cell surface. Okay, so it looks something like this. And what it does is these little triangles that are the antigens on the surface of the pathogen get taken by the infected cell and presented on the surface. So now your immune system can recognize that this cell has been infected. So we would call this an APC, which stands for an antigen presenting cell. Okay, so it's an infected cell. It's been infected by a pathogen, and the infected cell has taken the antigens from the surface of this pathogen and presented it on its own cell surface. And the reason it does that is so that a T cell can then bind to these antigens. So circulating in your body fluid, in your blood, you've got lots and lots of T cells. Thousands of different types of T cells. Now, the difference between all these T cells is that the receptors on them are a different shape. So you can see here, I'm drawing each type of T cell with a different shaped receptor. Okay, so each T cell has a different shape receptor on it. Now, simply by trial and error, these T cells will try to bind to these antigens that have been presented on this infected cell. So this process is going to take a little bit of time. But eventually what will happen is one that's a complementary shape will bind on to one of these antigens that's being presented by the APC. Okay, and when the right one binds, we call that we say it's step one, we call that clonal selection, because what we've done is we've selected which one of the T cells to make clones of. So we call this clonal selection. Okay, it's when a complementary T cell receptor binds to the antigen that's being presented on the antigen presenting cell. So this antigen presenting cell could be, for example, a phagocyte. Could be lots of different types of cells, but there's an example. So once this T cell has been selected by clonal selection, we say that it gets activated. So we could say and activation. And what's going to happen now is that this this T cell that's undergone clonal selection is going to be activated to divide by mitosis. So this one T cell that's got this specific receptor, not these ones with these receptors, but the one with the complementary receptor, is going to divide by mitosis. And we call that step clonal expansion. And what's happening is that it's dividing by mitosis because we want more copies of that specific T cell with that specific receptor on it. Okay, so we're going to make lots of T cells that all have the same receptor. They're going to be identical. It's divided by mitosis, so they all have, in this case, that kind of square-shaped receptor on them. Okay. Now we need them to differentiate because they're all the same at the moment, and we need them to differentiate into different types of T cell. Okay, so this next step is called, obviously, differentiation. Okay, and it's going to make different types of T cells. So some of them will become T memory cells. Some of them will differentiate into T killer cells. So M for memory, T for killer. Okay, so they're going to become different types of T cells. And the last one is called a TH, which stands for a T helper cell. So differentiation makes different types of T cells. Okay. So for the T memory cells, we'll cover that in primary and secondary immunity. And basically, these ones just stay in the blood for a long time, for years, circulating in the blood, in case you're infected with the same pathogen again, so that you can kill the pathogen faster. The T killer cells are going to go back. Let me get a different color. Let's get a highlighter, perhaps. These T killer cells here need to go back and kill any infected cell. So they will kill that, could kill that infected cell there, but they can go back and kill any cell that's presenting these antigens on. Okay, so when it says T killer, it's not going to directly kill the pathogens, it's going to kill the infected cells. Okay, so the T killer cells can kill, if you like, can kill infected cells. There's various ways that they can do that. And then we've got T helper cells. Now, what the T helper cells are helping is in the next video, but basically they help to activate B cells. Okay, B cells don't get activated straight away like these T cells did. They need some help, so that's what the T helper cells are there for. Now, one last thing that T cells can do is they can also produce something called interleukins. Okay, so the interleukins are kind of an extra, more complicated process of kind of influencing other cells. So the interleukins, when we look into the detail, are what actually help to activate the B cells, and the interleukins also can stimulate phagocytosis as well. So we do need to know about interleukins as well. Just a note on this video, if you're watching any old revision resources or books or anything, there used to be regulatory cells in this as well, but they have been taken out of the new specification. You do not need to know anything about regulatory cells. So if you see that, don't worry, okay? That's the end of this video. The B cells will be in a separate video, okay?